nice introduction. And it has been really nice to get to see you over the years and get to talk with you at MycoFest and at some of the other events. And yeah, I mean, since we first met, I myself as an educator have learned a lot, and my presentations have changed. I've made updates, and even now, as I'm sitting here, I could think of some updates that I would make. And so it's always a learning process, and I'm very grateful to be able to share this with the Central Texas Mycology Society. And like I said, the majority of the work that I'm interested in is really getting the education out there. The supplies are great, but in order to use the supplies, you have to have the education first. And so providing the education to the community, whether that's the local community or the community that's online, has really been the priority for me. And then from there, I've been able to create a business around it, which I've been very grateful to be continuing to doing. I'm now on year 4, which is very awesome. And I very much look forward to continuing into year 5 next year. So without further ado, this is my presentation from the field to the lab. And this is all about collecting your native mushrooms or your native fungi and propagating them or just collecting them for the sake of having these genetics in a database or in storage in one of your fridges, your freezers, or sending it off to a local university to their fungarium or herbarium. So the first thing I want to point out is in the photo, I'm pointing at a silver maple leaf tree. And inside of this tree cavity is this really beautiful lion's mane mushroom. And so this lion's mane mushroom right here has been growing consistently every December for the past 3 years. And so the state naturalist that works in this area will send me pictures every December and say, hey, look what I found again. And so I'll go back and take pictures, and each year the fruiting body changes, the size of it, how many fruiting bodies there are, the length of the spines, the coloration. So as you can see in my hand, just right below it, there's an agar plate. And this agar plate actually has mycelium. The same genetics that are on this plate came from fruiting bodies, not these ones, but the ones that were previous to the tree. And so I took back the genetics on the plate and took a picture with the new fruiting bodies that the tree was producing as a way to kind of demonstrate the variety that the genetics can produce in the outdoor environment. And then in the indoor environment, you'll notice there's a lot of differences when it comes to overall size, yield, growth rate, and sometimes even the flavor. So getting into the presentation, a bit of background of who I am and why I'm presenting today. I'm an outdoor enthusiast. I've always enjoyed being outside, whether it's playing sports or just hanging out, jumping on the trampoline with my family. Um, in 2018, I got an associate's degree in diversified studies. In college, I really didn't know what I wanted to do. My wife was um, my wife was going to school for nursing, and so I thought that that would be a good idea for me to tag along and go to the classes that she was going to. And it worked out really great right up until it was time to apply to nursing school. And that was when I realized that I probably wasn't cut out for that type of work. And my wife is a pediatric ICU nurse and has been for a couple of years now, and she's incredible and amazing, but that just wasn't for me. And later that year, I was hired as a lab technician working, testing soil, water, seeds. Eventually, when medical cannabis became legal, we started testing medical cannabis. And then in 2020, when the world kind of had this weird pause and changes, I was furloughed. And that was when I decided that I could either continue applying for jobs or I could try and start one around my hobby of mushroom cultivation. And so at the time, I was seeing a lot of cultivators online, whether it was TikTok or YouTube or Facebook or Instagram, post about growing mushrooms and selling them at their local farmers market. So I registered the Oklahoma Fungi as an LLC on August 10th of 2020. And as you can see, it took 2 years from starting the company to actually having the what I felt like was the skill set to provide education classes. So for those 2 years, I really delved into gourmet mushroom cultivation, and that was 2 years of in the making of a lot of failures, a lot of successes, but more so just the motivation to continue to learn. And so, as Angel mentioned, last year was our first time ever having a mushroom festival in Oklahoma. So, it was the first annual Oklahoma Mushroom Festival. We had 65 vendors. I think we had like 25 educators, roughly 50 volunteers, and we ended up having 1,776 tickets sold over a 1 day period. So, we learned from that event that the community in Oklahoma is very excited and very interested in mushrooms. And for that reason, of course, we're hosting a second event and continue hosting many more in the future. A big goal of mine later this year in November and December after the mushroom festival is over is also to come out with Oklahoma's first mushroom guide. So Oklahoma doesn't have an official mushroom guide or even an official list of poisonous mushrooms on our Poison Control Center website. So it's really a big goal of mine to help provide that information so our Poison Control Center can be updated, but also our citizens can purchase a guide that's not created by AI, that's listed on Amazon, that's from someone that lives here, that forages here, that works with local universities and other foragers to help identify mushrooms. So that's a little bit of background of who I am, what I'm really interested in doing. And this is more so on the company. So right now, the first thing I'll show on the right is our Mush Love gummies. So this is a lion's mane mushroom gummy that has 250 milligrams of full spectrum lion's mane extract. So we're extracting all of the beneficial compounds, the hericenones, arenosines, some of the beta glucans, and a lot of those other compounds that are beneficial. And we're inputting those into a gummy or into chocolate or into tinctures, depending on the product that we want to make or the product that you're interested in as a customer. Our number one product is the mushroom spores that we sell, or just mushroom genetics in general. And more specifically, the number one strain would be the Oklahoma Wild. So that's really awesome because that's a mushroom that I found on June 9th of 2022. And the fact that it sells more than a lot of these other commercial hybridized varieties is I'm very grateful because the opportunity to share the native genetics to places outside of Oklahoma has brought me a lot of happiness. And it's been so cool to see people just really support the landrace varieties, the native varieties. And I think everyone's very interested in cultivating native plants, and we should be just as interested in cultivating our native fungi. I also help with lab equipment, mushroom grow kits, mushroom ID. I lead private and public mushroom foraging tours, host the festival, and I'll continue down here, the author of the mushroom guide when that comes out next year. So the goal of today's presentation is to help you understand what it would take to want to take wild genetics to be able to isolate them and cultivate them in the indoor setting or the outdoor setting. So if you can think of your favorite mushroom, whether it's reishi, whether it's lion's mane, maybe it's wood ear, maybe it's turkey tail, maybe it's chaga. Chaga is not, you can't necessarily recultivate it. You can collect it, isolate it, and try to propagate it. But there's certain species that you're going to have better luck with. And the goal is to help provide you with the knowledge you need to get after those species in particular. And eventually, when you want to start experimenting, you can start doing that and keep me updated along the way. So collecting wild mushrooms, plants, or microbes for identification, cultivation, or to submit them to a lab for DNA sequencing purposes. So like Angel mentioned, the big myco blitz is coming up, and not every mushroom that we come across are we going to know the name to. And some of the mushrooms that we think we know the name to, when we run the DNA sequencing, turn out to be a completely different mushroom. So for me, this is always something that I find really fascinating is that mushrooms that look very similar, the genetics can be very different. And so the biggest thing that we can do as foragers is to upload to iNaturalist to collect the mushrooms and send them in for DNA sequencing to Mycota Labs or the Ohio DNA Mushroom Lab. Here's a quick list of 10 varieties that are native to Oklahoma or in a lot of other states and areas. All of these you'd be able to find in Texas and plus many more, but these are the ones that I have in culture that I've collected over the past 2 years. So that first one, Auricularia americana, is wood ear. Coprinus comatus is the shaggy mane inky cap. Ganoderma lucidum, which honestly, what we think is we don't have lucidum. We think we have sessile. So Ganoderma sessile, because the majority of the fruiting bodies lack a stem. So The fourth one being the lion's mane mushroom, Hericium arenaceus. Number 5 is the coral tooth fungus, Hericium coraloides. Number 6 is the chicken of the woods, Latioporus sulphureus. 7 is the morel mushroom, which if you've ever seen any of my videos, morels are my favorite mushrooms to forage, post about, cook about. And when it gets the beginning of the new year, the only thing I'm thinking about is how many days until morel season. So the Morchella americana is the same yellow morel that you guys would be foraging down there in Texas. Number 8 is the Pleurotus austriatus. This is a delicious oyster mushroom. Now, depending on if it's spring or fall, sometimes you can find different varieties, but most commonly we're finding the austriatus species. Of course, you can find Psilocybe cubensis in Texas, in particular North Texas, especially around the Red River in between Oklahoma and Texas. There's a lot of mushrooms that grow in that area, a lot of cow pastures as well. And last but not least, turkey tail. If you've heard of turkey tail mushroom, it's a great mushroom. It's abundant, and it's one of the easiest that you can find, collect, and get into your culture bank. So that's 10 species that I've collected over some time. I didn't do this all in 1 month. And these are some of the varieties that you can have great luck with. And all of these varieties that are listed are delicious edible species. Some of them, of course, like number 9, of all of them does have psychoactive properties. However, it is still edible. Just proceed with caution. Speaking of active mushrooms, this is what the mushroom looked like that I found in Oklahoma on June 9th back in 2022. So this just kind of ominous, just very interesting, kind of nonchalant looking mushroom is an active mushroom. This is a Psilocybe cubensis mushroom. And it's really, I think it's about 7.5 centimeters tall. So it's quite short and it's a very, very small mushroom in particular. And so coming across this, I was quite surprised because it was the first time in 3 years that I had been foraging and had come across it. And so from that one day of finding the mushroom, collecting the spores, and then passing them along to friends, as you can see from the left, what it looks like in the wild, and then over here on the right, what it looks like when it's been cultivated indoors. So this is just a really great example of what mushrooms can do, how they can change when they go from their natural environment to their indoor environment, when they go from these very rapid changes in humidity, temperature, and the constant exchange of oxygen and CO2 outdoors versus indoors where there's artificial light, restricted airflow, and the supplemented nutrition. So very, very, and there's also lack of microbial diversity. So there's a lot of factors that go into mushrooms, and the way that they look is very much dependent on where they've been collected and what they first looked like. And from there, you can select phenotypes that you particularly like. And so this over here on the left is what we call Oklahoma wild. And over here on the right, this is what we call Oklahoma wild footlong. And that is because they produce footlong or foot tall mushrooms. So very, very interesting and unique variety. And of course, if you're interested in this, you can find it on the website. And definitely stick around to the end of the presentation. We're going to be doing a giveaway for those who can come to the Mushroom Festival. We'll be giving away 2 tickets. And for those who know they won't be able to attend but still want to participate, I'll be giving away 1 free spore syringe of this strain right here. So stick around. We'll be giving that out. And I really look forward to helping spread the spores down to Texas. Here's some more great photos of what it looked like on that day when I was collecting the specimens for the spores more so. So over here on the left, you can see this over here where my point mouse is. These mushrooms are the non-active species LBM mushrooms, little brown mushrooms that are hard to identify. But this pin, this pin, this pin, and this pin, as well as this one, are all the primordia of the Psilocybe cubensis. Over here, these mushrooms on the left-hand side of the photo, those are also what we'd consider LBMs or some smaller inky caps if you wanted to get into the ID. And over here on the right, as we can see from the bluish bruising and the mycelium down here, as well as the spore deposit on the cap right here and the black skirt, this is a fully mature Psilocybe cubensis. And my favorite photo that I've ever taken would be this one right here in the middle. This is a Psilocybe cubensis mushroom and 2 pieces of horse manure that the mushroom is growing in between, and it's actually stretching out in between and collecting the nutrients from both of them to be able to grow. And the reason that it's lost its orange coloration is because it's been sun bleached. So it's been sitting out open in the field with no protection. And that oftentimes can make them hard to ID. But because I was in the area and with my experience in foraging, I was able to ID this, and it's definitely something really cool to see all different stages from pinning to producing spores to fully mature all in one day. Now to get started on what you're going to need when you want to go out into the field and then come back to the lab. So number one would be a bag to carry all of your materials. Of course, you could wear a cargo vest and cargo pants, but you don't necessarily want to have everything in your pockets because when you're bending over or grabbing mushrooms on the forest floor, something may fall out. So as a forager who's lost many foraging knives, unfortunately, the number one thing I can recommend is that you get a bag and you take everything and you put it inside the bag, and each time you stop, you open it, collect your mushroom, and put everything back in there. Number two is to go out and actually find some mushrooms, right? Once you have all your materials, now you need to find a mushroom. And you can choose any mushroom that you find, but in particular, the mushrooms that I'm looking for are edible mushrooms. So if I can find a mushroom that's edible, whether it's for its culinary value or any of its other properties, this is something that I would have preference over than something that was just growing in my front yard. So from there, the one thing you want to have on number three is your phone. You want a fully charged cell phone, not just in case of emergencies, but you also want to be able to download the iNaturalist app so you can post about your observations. And one thing I want to point out about iNaturalist and the observations that are on there is that when you were to look at them, these observations aren't the most common mushrooms in your area. They're the most commonly posted about mushrooms by humans that find them interesting. So there's a lot of mushrooms that humans that we're passing right by, that if it doesn't catch our eye and if it's not interesting enough, we're not going to post about it. And that's, that in itself can kind of skew the data, because when you look at iNaturalist and you look at the most common species ID'd, you may think that that's the most common mushroom you'd see. And so, sometimes it is the most common mushroom you see, but other times you'll notice that the species that are in the top 10 species are not always the most common. They're just the most commonly observed, and posted is more so the important part. Number four is to collect or to purchase or to even make your own sterile agar plates. You can use any recipe you like. I like to use this recipe is potato dextrose. I'm sorry. This actually is MEA MEA agar with a little bit of blue food coloring, just because I like the way that if you look at the right side of the agar plate, it's kind of lost its blue color. And that's because as the mycelium digests the nutrients in the plate, it loses its color. So it's a really cool way to have an indicator of the mycelium. And that species in particular on the plate is the lion's mane mushroom, Hericium erinaceus. And it's a very thin filamentous mycelium that's not necessarily as distinct as something like reishi or oyster. So by putting a light blue color and then that light blue color changing as it grows, it's able to help me see that it's an indication that the nutrition is being digested by the mycelium. Number five is very important, and that is to actually have something to wrap around your plates once you've put a collection on there. So parafilm is very commonly used. It's like lab saran wrap. You can also use a plastic Ziploc bag or anything else that you think would keep that lid on that Petri dish without it opening up and getting contamination inside. Number six is a scalpel for tissue transfers. So if you're doing the cloning of the mushroom, you definitely want to use something, whether it's tweezers or a scalpel, something that you can flame sterilize. And that is what number seven is about. So if you have a lighter or a torch or a bactincinerator or a flame induction burner, all of these are great things that you can use to sterilize your blades. So that way, when you go to do the cloning to try and isolate the genetics, you're going in with a clean blade and a clean tool, much like they do in the medical field. Number seven is, I'm sorry, number eight is a Sharpie to write the name and the date. Now, this is very important because if you're growing multiple strains, you definitely don't want to get them mixed up. I've heard from numerous times. I was actually just talking with a friend 2 days ago who purchased lion's mane genetics from a vendor, and when he grew the genetics out, it was actually a chestnut mushroom. So sometimes things can get mislabeled, sometimes things can get mixed up, and the one way to keep that from happening, especially in your own genetic category, is to label it as soon as you fill the plate and wrap it. So after you're done with the transfer, you've sealed your plate, write the date. You can even write your name, or if you have multiple lab assistants, or depending on your setup, you can write your name, the date, and the species. So that all of these things will be a great indicator of what what is in the dish, who made the dish, and when the tissue was first placed on the dish. Number nine is something that is optional. You can take the mushroom field guide into the field if you want, or you can just use iNaturalist for the observations. And then when you get back home, you can use your field guide to look and re-update the information on iNaturalist. So for me, the mushroom ID, the mushroom guide, taking it out in the field is something that I typically do on my public foraging tours. When I take people out and show them how to ID mushrooms, I'll take the guide. But when it's just me going out there, I typically don't take the guide. I'll just take really good photos of the top, underside, the mushroom in its natural area. And then from there, I'll upload the observations and I'll go home and I'll compare my photos to the guide. So that way I'm not lugging around the book around with me through the forest or potentially getting it dirty because the book that I have, the pages aren't waterproof. Number 10 is a bottle of water and hiking snacks. If you're out there in the field and you depend on spending a lot of time mushroom foraging, or you end up coming across a really great haul of mushrooms, sometimes the days you think that you're not going to find anything, you find the most mushrooms. And the days you think you're going to get out there and find a lot, you find just a little. So it's always good to have a bottle of water and hiking snacks with you just to keep you going, keep you foraging if you can stay out there. Number 11 is a clean space to work at home for your agar. Behind me, as you can kind of see, I'm working. This is a 4 foot by 2 foot lab. So I'm in my garage right now. And because it's my garage, I don't have a clean space. So in order to have that, I've got a grow tent and I do all my lab work in the grow tent. And I have this really nice flow cube. So this is a 2 foot by 2 foot flow hood. And if you're interested in flow hoods, I can give you a discount code. You can send me a message on Instagram at Oklahoma Fungi. You can also send me a message via email, oklahomafungi@gmail.com. If you're interested in these, I can help you. If you're also interested in what's known as a still air box, I don't have the still air box behind me, but a still air box is a really great option that's not as expensive as the flow hood. You really don't need this unless you're really trying to get into commercial cultivation and growing at a scale in which you're selling at the farmer's market or to your local grocery stores. So if you can reduce your cost and keep your capital really low, I would definitely get a still air box. And I can also recommend you a discount and a company to get those from if you're interested. Getting into the agar and the many different types you can make. The photo on the right shows what it's like to pour a little bit of agar. In college, a professor told me that when you're pouring, right when the plate looks full, you should stop. Of course, that is just very general, and that doesn't really help depending on the depth of agar you want. It takes a long time to get that pour right. Sometimes your plates will be twice as thick as the one below it. And sometimes you think you're going to pour 100 plates and you only end up with 85 because you poured so heavy. So it takes time. It also takes practice to make the agar. There's water agar, grain agar, potato dextrose agar, and light malt extract. So these are all really great options, as well as the additives, peptone, yeast extract, food coloring, and activated carbon. The activated carbon is really fascinating and really cool because it turns the agar black. And as we all know, that mycelium being white, when it grows, it has a very, very beautiful contrast. And I have some photos coming up of what that looks like. So once you've isolated your culture and you're starting to grow it out to be able to recultivate, the goal is to decide what type of mushroom, of course, that you can always ID the mushroom once it's fruited, but when you transfer the genetics to a bag, you can see the bags in the photos This one kind of, I think, has a date right here, but the two other bags, they don't have the date and they don't have the name of the species. So when you're growing in bags, it's important to take a Sharpie to write the day that you inoculated it, the day, I'm sorry, the day you inoculated, the species that it is, and this will help you keep track of the growth rate. So when you're growing in bags, the benefits are that it helps with fast colonization. And this is the start. This is what's giving your mushrooms the nutrition that they need to go on to the next step, which would be fruiting. So if you're wanting to cultivate in logs, instead of putting grain bags, you would be growing on these little wooden dowels. And I'm sure the Central Texas Mycology Society has done a class or at least talked about that because the logs is something that's really, really great to do. Although it has been a very hot summer with very little rain, it's a great way to be able to grow mushrooms when you have a bunch of extra downed wood or logs that you can use to regrow some mushrooms. Number two is when you're growing in bags, oftentimes you need some LED light or some light indoors to be able to see. Make sure you have no contamination. Number four, growing in bags is also a sterile environment. So as long as your bags are clean from working in that still air box or in front of a flow hood, there will be no contamination and you'll be able to tell because those bags are clear and you can turn them all the way around and make sure that you don't see any discoloration. And if you do, I recommend taking a Sharpie and circling the contamination or what you think might be contamination. And the next day, go back and check it. And if what you think is contamination has grown and it's bigger than the circle you drew the day before, it most likely is a potential contaminant. That is also possible, mainly because of when you're working in a very sterile environment, anything that lands inside that bag is going to want to eat the nutrition, eat the grain that's inside of it. Another great thing about the bags is that depending on how you use them, they can be reusable. They're great for optimal moisture content. So as you can see in the photos, there's a bunch of humidity at the top of the bags. It hasn't escaped, and that's great because we want to keep that moisture inside the bag so our grains are nice and hydrated, our mycelium can thrive and continue to grow. These bags have these nice big filter patches at the top. These are really important so that way the CO2 can be released and your mushrooms can breathe in oxygen. Just like we breathe in oxygen, the mycelium and the fungi also breathe oxygen. So very, very awesome, with plants and fungi creating this circular air exchange. It's perfect if you're growing any plants indoors right next to your mushrooms. Number 8 is the bags are clear so you can see the mycelial growth, which is a very, very good thing about the bags. As you get into the commercial scale, you might notice that some growers use black bags, but what you'll find is that even if they make 99 black bags, they'll still make one that's clear. So that way they have an indicator bag of what all the other ones might look like. So if the indicator bag is contaminated, that might mean all the other bags are contaminated. But if the indicator bag is clean, then most likely all the other ones are clean as well. Number 9 is some of the bags are biodegradable. So depending on who you purchase your bags from, some of the bags are, when exposed to UV radiation and heat over time, can biodegrade. And number 10, a reliable colonization schedule, as long as you're putting the same amount of grain in each bag. If you put 1 pound of grain in one bag, 2 pounds of grain in another, and 5 pounds of grain in another, the grain bag that will colonize the fastest is the one with the least, the smallest amount of mass. So the one that only has 1 pound will colonize much faster than the 2 or the 5 pound. This is an example of what log cultivation looks like. So number 1 is Mother Nature takes control. So when you cultivate logs, what you're doing is inoculating these tiny little holes that you've drilled with a 1/4-inch drill bit and putting these wooden dowels into the logs. And so after that, you can kind of see all this white yellowish stuff. This is typically beeswax that's used to seal the holes. That's to make sure that no other contaminants can get in and infect the wood before your mycelium, your shiitake mycelium, has a chance to colonize and start fruiting. So number 2 is that this cultivation method only takes up space outdoors. So if you live in a house and with a bunch of animals or kids, and you don't want them to be messing with your mushroom hobby or your mushroom things, and you want to cultivate outside, this is a really great option because it produces multiple harvests. It can help spread the spores around to other trees in the neighborhood, also just other trees that are in your backyard that you've already inoculated, to make sure that they're myceliated and will produce mushrooms. It's almost a maintenance free after inoculation. You do have to dunk them in water to cold shock them and bring them back out. So that is something that number 7 talks about. If you cold dunk, it does help to encourage what we call a snap, and the mushrooms will begin to grow. And then number 8 is that after a winter storm, you can find a lot of these hardwood logs. And I would recommend cutting them from 3 foot to 4 foot sections. And then you can stack them in the backyard. And that's a really great way. You can lean them up against the house like this one has in the photo, or you can stack them depending on how much space you have. So growing in tubs. So if you've ever heard of a monotub, that's what these are right here. Each of these tubs is a 66 quart clear tote that you'd find at Walmart. They're stackable, which is great. They're plastic. They're cleanable, reusable until they crack and break. So really, really enjoy working with these. You can grow gourmet mushrooms. You can grow many different types of mushrooms in these. So I'm going to go over some of the benefits. One of them is that you do, or the go over the specs for growing in tubs. As you can see, provide LED lighting. It doesn't necessarily have to be LED. Up here, we can see, I think it's called an incandescent bulb. So if you have LED strips, I would lay the LED strip across the top of the tub. But if you only have one light like this or a closet light, one of those is just enough to help give your mushrooms direction. So mushrooms and fungi don't photosynthesize. They don't create energy from sunlight. But what they do is I think it's called phototropic, which is where they use sunlight for direction, kind of like a sunflower. If you notice, sunflowers will follow the path of the sun. This is actually, I think it's called heliocentrism. Yeah, heliocentrism is the ability to track the sun's movement or to follow the sun. But in this artificial cultivation setting, they're not tracking the sun, they're just following the light, growing towards the light. Number 2 is the tubs. The tubs are stackable, as you can see, reusable until they crack. Now, of course, when you stack them, depending on how heavy they are, over time, you may find that the lids crack more so than the bottom. Now, creating these also is a sealed, sterile environment. You can see that there what looks like cotton balls on the sides of the tub. So you take a 1/4 inch drill bit, and when you're drilling these holes at the top, it allows for air exchange. So if you look right here where the cursor is, everywhere where there's that little cotton ball, you'll notice that there's a larger circle around it. And that is the humidity and the CO2 that is leaving the tub. So it's actually exiting through this little circle on the circles on the sides, and then the oxygen is coming in. So this is what we would consider like a low-tech air circulation system. We've also loved the tubs because they're clear. So in almost every tub in the photo, you can see the mushrooms growing inside of them. The only issue sometimes is that humidity does build up, and that can sometimes block the view of the mushrooms. Also, with number 7, not every species can you cultivate inside of a tub. So some species are going to be better cultivated, better suited for outdoors or for logs or for just your general hardwood block that you guys are, the Central Texas Mycology Society helps when you guys are giving out the composting blocks, those exact same ones that you can take and put in your garden or to regrow mushrooms outdoors. Those blocks you can put inside the monotubs and you can fruit them out as a very small humid chamber. So a lot of different options for growing in tubs. Now getting into the actionable steps. The first one would be to gather tools and make a game plan. Make sure you have the updated iNaturalist app and you have the most up-to-date Google Maps. So that way you can save the coordinates. You can also make sure you know where you're going and how to get back. Number 2 is find a fresh edible mushroom to clone. So in this photo, this is actually a second time. This is the third year I found this lion's mane mushroom fruiting. So this is the same tree that you saw in the photo at the beginning of the presentation. But as you can see, there's this very large fruiting body at the bottom, which weighed about 8 pounds, and this smaller one, which is about 2.5, 3 pounds. So I have my mushroom field guide right here, and I'm comparing it to the bear's head tooth, the coral tooth, and the lion's mane. And comparing this illustration to this fruiting body, it's very easy to tell that this is the lion's mane. So it is very, very great because when you're finding this mushroom, not only is it delicious, also very good for the brain. And when you have your field guide, you can ID it on site. Up next is to upload the photos and all the description information that you can to iNaturalist. So this is a nice little screen grab of what iNaturalist looks like. This is all the fungi at the time. I think I took this screenshot last year. So as of right now, iNat for fungi has 5,588,172 species. This information I'm sure is outdated and it's been updated, but shout out to John Plisk, who has 419,893 observations of fungi. That's almost 500,000 observations. So that's just really, really awesome to see people getting out there and really doing everything they can to help promote fungi, to help bring awareness to the observations, what they are, where they're growing, and helping to educate the community on what our biodiversity looks like. Number 4 is decide if you want to clone the mushroom in the field or if you want to take it home and clone later. So in between, this is I forget what the type of tree this was, but this is beautiful Pleurotus austriatus. This is the oyster mushroom. And in between the tree trunk that's been decaying and the bark is this perfect little microclimate. And a microclimate is a small area that has a different temperature, humidity, and lighting conditions, maybe even CO2 oxygen conditions than the greater area. So the area over here to the right of the mushroom is exposed to the sun. There's little to no shade, and there's no way to create humidity. But in between the bark and the tree trunk, there's just enough space for a microclimate to develop, which is going to really, really help to propagate these mushrooms to help know that the fruiting conditions are optimal for them to grow. So this is a really great day because these oyster mushrooms were popping right out of the bark. Up next, if you decide to clone in the field, your culture will likely need 2 to 3 transfers before it's a completely clean isolation. So this tree is the same tree that I was cloning the lion's mane from, and you can see the tiny piece of tissue inside the agar plate. Now, of course, because as you can see, the plate is out in the open in the air. There's no flow hood. There's no still air box. The odds of bacteria or other spores falling onto the plate and growing at the same time is very high. And that's why you'll most likely need 2 to 3 transfers of your selected culture to really isolate it on the plate and make sure that you have growing is what you think it is. There's been many stories of people, like I mentioned earlier, even my friend who purchased a culture and it ended up being something different. I've had friends collect cultures out in the field. They go home, they isolate what they think they collected, and next thing you know, they're growing a completely different species and they don't have the genetics that they thought they did. So, going out there, it's all a learning process. And I recommend taking the mushroom fruiting body home and doing the cloning at home because you have a higher likelihood of getting a clean culture faster. So if you can harvest the mushroom and clone it back at home, you'll have a lower risk of contamination. In this video, this has since been taken down because YouTube deleted my channel for drug abuse, which, as I'm sure many of you know, majority of the social media accounts, the companies, aren't very fond of mushrooms. So I'm doing my best to promote the education on platforms that allow it. YouTube, I was doing a lot of videos. I had over 140 videos all about education and foraging. And unfortunately, they just took them all down. So this is a screenshot that I still had from one of my videos, which was going into your local Walmart, purchasing all the mushrooms that they offer, and then showing you how to isolate the genetics and then recultivate those same species at home. So you never have to go out and buy them again. You can continue to regrow them. You can learn about a new hobby and grow your food sovereignty. Step 5. Once you have an isolated culture, expand it to make more agar plates so you have backups. Expand it to a liquid culture jar, which is this what's right here in my hand below the agar plate, and or inoculate a bag of sterile grains or wooden dowels. So this is a grain bag, but you could also do wooden dowels if you're going to be inoculating logs. Number 6, the substrate in the container that you use are dependent on the species you want to cultivate. So if you want to grow oyster mushrooms, you can also grow them on straw, which is really great using a low-tech cold water pasteurization with a little bit of hydrated lime, or you can use hardwood blocks. So the hardwood blocks take sterilization, or you can go the low-tech route, which is a great option. So depending on which mushroom you want to grow, not all of them will grow on that hay low-tech option. Sometimes you will need to create that fruiting block that's been semi-pasteurized or even sterilized to really make sure that it's everything that's in there that could contaminate your grains doesn't. So the most common way to cultivate mushrooms indoors is to grow on hardwood pellets. In this bag over here on the left, you can see these tiny little pellets, and these are also what people use for grilling. So when you grill, you have these little hardwood pellets that you throw in there, and these help to burn more evenly. These same mushroom pellets are great. I'm sorry, these same hardwood pellets are great for growing mushrooms, but you do have to be careful about the ingredients. If you're purchasing hardwood pellets that have been added with have what they call flavor additives, like a cherry or an apple, or I'm trying to think of the other. There's a couple other, I think, like maple. Of course, using different hardwood species, trees, is just fine. You just want to make sure that they're not adding any artificial flavorings or additives to the hardwood pellets. And that's mainly because what you feed your mushrooms, they digest, and then you eat your mushrooms and digest. So what you put into them goes into you, just like all the other food we eat. And that's why soil health is so important. And fungi are key to making sure our soil health and our gut health, our brain health are in where they should be, which is happy. So, the two most popular ways to cultivate manure-loving mushrooms is indoors, inside bags or modified tubs. So this is what a mushroom looks like growing in an all-in-one bag. And over here on the right is what they look like from a bird's eye view growing inside of a tub, a monotub. Step 7 is to inoculate your substrate with colonized grain or wooden dowels, depending on the method that you choose. So the photo on the left shows someone taking myceliated grain and expanding it to grain that has yet to be colonized or yet to be myceliated. So they're expanding the genetics so that way they can grow more mushrooms on more substrate. On the right, we can see this wooden log and these tiny little wooden dowels that they're taking the hammer and they're knocking into the wood, and they'll go back with this beeswax right here and seal each one of these holes. And if you can see closely, there's these little tiny brown pegs sticking out right here, and that's what they look like when they're knocked inside the hole. Step 8, place the bag or log in optimal conditions to allow the mycelium to expand throughout the substrate until it's fully colonized or myceliated and ready to start producing fruiting bodies. So the mushroom life cycle right over here is typically what we all think about. But from this stage on the grains, what we're really trying to do is give it the right conditions to form primordia and then eventually produce the mushroom. Step 9, once the mushrooms are fully grown, harvest the mushroom that you like the most for its color, its size, its texture, its taste, its yield, or how fast it grows. Keep in mind when you're collecting these native species, these wild mushrooms, these mushrooms typically are going to grow faster. They're going to be larger. They're going to have a more concentrated flavor. The texture may be even a little bit different. And of course, the growth rate is going to be different. And that is because outdoors in nature, these species are having to compete with hundreds, if not thousands, of other microbes and other things and variables that are trying to combat them to keep them from growing. So in nature, nothing is sterile. And these mushrooms are fending and essentially fighting for their lives to grow and reproduce. And so when we grow them indoors, what we want to do is select the mushroom that we like the most for whatever that may be. And so if you're looking for overall yield, don't select the tiniest mushroom that grows, select the largest mushroom. If you're looking for the tastiest mushroom, that is something that you'll have to harvest the mushrooms individually and taste test them, maybe do a little cooking, sampling, and see which one in particular you think has a better flavor. Now, of course, it's not going to vary that much, but some of them will vary depending on the size of the cap, the size of the stem, things like that. Number 10 is clone your chosen mushroom, regrow it on agar, transfer to grains, and eventually spawn to substrate to grow more mushrooms. So this is a nice agar plate that is, you can see the isolated culture growing on. It's a beautiful contrast with the black background from the activated charcoal and the white mycelium. So we know that the culture is clean. This is a really nice photo with the microscope of what that mycelium, that hyphae looks like expanding. So from here, we know that we can start to grow more mushrooms that are fresh and healthy, and that it's the culture we want. Now, when you're refruiting the mushrooms that you've selected, once you've selected for size, you want to continue selecting for size. If you've selected a mushroom for its growth rate, don't select the next time for something the complete opposite. You really want to try and stay selecting something continuous. And of course, your wants and desires may change as a grower, but the goal is that each time that you select something that's outside of the wild genetics you collected, you're going to be kind of narrowing that genetic pool. So keep that in mind, is that if you want something that's going to produce a variety of different expressions, you want to work with the landrace genetics that have not been hybridized, that haven't really been regrown. Step 12, once you're happy with your selected variety that you're growing, no matter what if it's for its size, flavor, texture, yield, growth rate, the first thing you want to do is make some agar plates, some liquid culture, or some agar slants. And these you will be able to take your mycelium, add onto those plates, and then put them into cold storage. Now, cold storage is very important because if something goes wrong inside the grow room and something contaminates, you can always go back to your cultures and have a clean culture to use. You know it's the same genetics you were using at the beginning. You know they're clean, and you know it's going to be consistent. So that way, each time you try and regrow the same species, it's actually the same variety. The same gene pool. So some things you should know about wild forage genetics. This photo is a really nice log that has oyster mushrooms fruiting on it. That's probably close to 10 pounds of oyster mushrooms. So that'd be a really great haul. Number one is that they're acclimated to non-ideal temperature, humidity, and lighting conditions. So out in nature, nothing is ideal, nothing is consistent. And the mushrooms that are growing indoors inside the lab that have LED lighting, that have the fans for the AC and the CO2 and the oxygen, all of these things typically in nature aren't even a factor. So in nature, they have to grow fast because that's the only way that they'll survive. So they typically grow faster than your commercial strains. And even as a breeder and as friends who do breeding, what we'll notice is we'll find companies that will take a hybridized variety that grows really well indoors, and they'll cross it with a variety that grows really fast outdoors to try and create something that grows fast indoors, but also a great yield. Number three is that these wild varieties can typically be more resistant to bacteria, molds, or other contamination. They can require more fresh air exchange because what they're used to is that constant ability to breathe in, breathe out with no filter, no restricted access to airflow. So when these mushrooms are grown inside a grow tent like the one I'm in, they don't have as much access to to breathe in that oxygen and respirate that CO2. Number five is something that I mentioned earlier, which is that they're commonly used to breed new strains. Something that's really cool about mushrooms and just DNA sequencing in general is that new mushroom species are being discovered every month. If you've heard of Paul Stamets, which I'm sure many of you have, this was a mushroom, Psilocybes stametsi, that was named after him, or in a sense, as a way to honor him and his work, his contribution to the mycology community. So this is a really cool mushroom. It looks like a nice little witch's cap. So the really pointy top and the cap kind of unfurled in a very perfect way that's good for Halloween. Up next, we have DNA sequencing. Mushroom species is better than ever before. This is a photo from one of Alan Rockefeller's DNA sequencing workshops. I don't think he teaches these anymore, mainly because the accessibility to testing has become so much better. We can actually send out samples to the Ohio DNA lab and get anything that we can find as long as it's on iNaturalist DNA sequence for free. And same thing with the Mycota lab when MycoBlitz comes up. Speaking of the Ohio DNA Mushroom Lab, this is a nice screen grab from Kyle Cannon's website. This is all about the work that he does out in Ohio collecting mushrooms, DNA sequencing, and it's all for the betterment of the community. So definitely check out his work. Definitely check out the Ohio Mushroom DNA Sequencing Lab. That's it. Thank you all so much for your time, for your interest in the subject. I hope that I was able to inspire you, to give you a little bit more knowledge on what to do when you go out foraging, what you might need if you want to start collecting genetics, what you might do once you have the genetics and you want to regrow them, and how to store the genetics for later or to distribute them out to friends. I hope that it was informative. I too much. And if you have any questions, of course, you can reach out to me. My email is oklahomafungi@gmail.com. My Instagram is oklahomafungi. The festival is okmushfest.com. And that is this weekend. We are I think we're 4 days away.
Yeah.
And this event is the biggest celebration for mushrooms in our state. And I really encourage everyone to come up here if you have the budget, if you have the time. We know that it will be well worth it, and we'd be very excited to have you.
Yes. Well, with that, should we do the giveaway and then we'll get to Q&A?
Yes, I love it.
Okay, there's a few last minute people showing up just in time. So we're
Perfect. I'll
about
let
to
you.
do the giveaway. So, Jacob, I'll let you do the trivia question and I'll watch for the chat. So the first person to put the correct answer, get your QWERTY, get your keyboard warmed up, get your fingers on your keyboard.
Get ready.
Get ready because the first person to type in the correct answer will be the winner of 2 tickets. And if you want to forego tickets, Jacob has got that native wild spore syringe, the wild Oklahoma wild genetics. Does that sound good to everybody? Yes, applause all around. Okay. All right. Yeah. So does everybody got that? So you can forego the tickets if you're unable to make it and take the spore syringe, and we'll find somebody that can go to the festival. Because we have some folks here in Austin that really want to go. We'd love to give them tickets. But yeah, so get your keyboards ready. So here is our question. Go, Jacob.
Okay, so if you're familiar with the Devil's Cigar or the Texas Star, it's your official state mushroom. This mushroom is very rare, but it has also been found in Oklahoma. And the first person to type the county that this mushroom was observed in the chat wins 2 tickets to the second annual Oklahoma Mushroom Festival. So I'll repeat the question. Which county was the Devil's Cigar or the Texas Star, the C. Giaster mushroom? Where was that found in
Greg, snap,
Oklahoma?
snap, snap. Greg came in. First
Can
answer was correct. Choctaw County. Woohoo. So, Greg, can you
Amazing.
make it to the festival or do you want to take the spore syringe?
Sorry, too many mute buttons. I can't make it, unfortunately. I would love that syringe.
Whoop. All right. Well, we'll get your contact info. You can message Jacob. You have his email, right?
Yep.
And give him your address. Jacob, that was so great. Greg, good job. So Choctaw County, our Texas Star mushroom started popping up in July. And I say "our," not that I'm like claiming it. I'm like, it can be in Oklahoma too.
You can have it. It's fine. You can have it.
We're trying to clone it in the meme lab, which they're having their lab night tonight. But we're trying to clone it again. It's been cloned before, and we're trying to get some metabolomics and understand the chemistry of it, because so many people want to know what's in it. You know, it's the only mushroom in its genus. We're going to be adding walks to go look for this hissing. Y'all, it hisses. This mushroom hisses when it spreads its
Yeah.
spores. We've witnessed it now and recorded it out in the wild, go on our walks. So if you're not in Texas and you're in Oklahoma or in the northern counties of Texas, it's abundant, like in our region, but only found in sort of the I-35 corridor, and then Japan, and then Eric Cho,
Taiwan.
the mycologist, also found it in Taiwan. There's some mycologists there that found it, and they're trying to get like the official scientific records updated. Um, but anywho, so
Let's go
yeah,
for the second one.
yeah,
Second second giveaway. So whoever can send in the Latin binomial, the two-part name of the yellow morel, the mushroom that
hmm,
is the most prized in our area in that March to April season. So two part the Latin binomial of the yellow morel. First person to put it in the chat, if you can come to the mushroom festival, you get 2 tickets. Let's see
boom,
if
boom,
anyone can
boom.
get it. And keep in mind, it is not the European version.
Nope, not
Some
Irina, not the European.
very close, very close. Let's see if she can scoop it.
Turn it around. Flip that continent around.
Mm,
Those spores adapted once they floated across the ocean. Yes,
yes,
Tina. I like with two eyes. All
amazing.
right. Well, we've got a few questions left in the chat, and feel free to raise your hand and come unmuted. So Donna was asking, what are regular crimini mushrooms grown on commercially?
Most of them are grown on a mixture. Typically, it's a outdoor windrow compost. So they're taking compost from a lot of the cattle farms. They're amending it with locally sourced straw or hay, and from there they're going through a natural pasteurization process that is then inoculated with grain. And I've actually been on a tour of the largest button farm in Oklahoma. They produce 28 million pounds of button mushrooms every year. And so these farms have just these massive football fields, size fields that are filled with composting rows. And so they're growing on this manure and straw-based compost that is then supplemented with the grain spawn that's pre-myceliated. And from there, it's the natural pasteurization that occurs kills off the negative microbes and allows just enough time, space, and nutrients for the good ones to start growing.
Mhm. Yes. Okay. Let's see. Anybody else want to raise their hand? Have any questions? There was Greg, I think you had a question. Yeah. So Greg, do the mycelium cultures growing in the dishes require light at that stage, or is that only for fruiting?
Yeah, at that stage, the agar plates can be kept in complete darkness after you've done the transfer. You can also do the same thing with your liquid culture. It doesn't need to be in the light. However, bringing it into the light to view it, make sure that it's clean is the best thing you can do. Yeah, so definitely want to make sure that, if you can keep the lights off, you can do that. When it's fruiting, you want to keep the lights on, but when it's on the agar plate, when it's in the grain bag, like I actually have a this bag right here is some mycelium growing on grain. And so this bag, whenever I leave my lab, I just turn off this light above me and it just hangs out in the dark. So the agar plates and the liquid culture don't need to have light all the time. It's only when the mushroom is fruiting.
Okay. Irina asks, what's the common mistake that new growers make?
I think the most common mistake is being unsure if they want to, if they have the skill set to grow. I think a lot of times when people get into mushroom growing, they're like, "Oh, it's going to be like a plant," and plants require sunlight and water in a way that's different than mushrooms do. So, I think oftentimes we kind of go into growing mushrooms as we grow plants. And even though you can grow both and even grow them together, they do take different methods. Some of them take different nutrition, and so that can be some of the reason that some mushrooms will produce better than others will.
Yeah. Okay, so Donna asks, why do shrooms need light to fruit? I love this
Well,
question. I love that
yeah.
answer too.
Yeah, well, the interesting thing is they don't necessarily need them to grow in the dark. So if you could grow mushrooms in complete darkness, but the really interesting thing is that when you do that, they don't gain any color. They stay white. So if you've ever eaten the enoki mushroom, it's a really tall, skinny white mushroom. And these mushrooms out in nature are orange and brownish and very beautiful. And sometimes they can be a little rare to find. But when you grow them indoors and in complete darkness, they're completely white. And so it's not necessarily a bad thing. They don't lose their nutritional content. They do kind of change their shape, their form. They may even change their color, but for the most part, it's still the same species, the same benefits, just growing in different conditions.
Yeah, and the melanin mushrooms and plants, have
Hmm.
melanin in their skin like our skin, and that is
Hmm.
how they absorb that UV light. But I'm curious because there was that study, and of course, like, as Paul Stamets, I think it was on fungi.com in a study that they commissioned where they exposed store-bought mushrooms to UV light after they were even harvested, and they were able to measure that the vitamin D content was
Hmm.
300 times more. So that people have always told me that, and I'm like, well, I guess that technically would make like more like wild mushrooms, like mushrooms that are growing in like dapple forest light, or like
Yeah.
mushrooms in a farm that are exposed to UV light. They have those nutritional benefits, but I'm also just kind of like, I'd love to see more studies of that, and just like at different kinds of species too, because the different mushrooms, like the darker the mushroom, the more melanin, the more vitamin D. Wood ears being one of those that has some really wild studies on melanin and the benefits of, you know, consuming it for its melanin.
Yeah, I'll be honest, I have been hearing a lot of people talk about, putting your mushrooms out, but I don't know a lot of people that are doing it. I've met
Mhm.
one farmer in the past 4 years of traveling all around the country and touring farms that actually takes their mushrooms, and not necessarily when they're selling them fresh, because when they're fresh, they take them, harvest, and put them in the fridge. But when they're making products with dried mushrooms, they'll actually take their mushrooms and they'll put them on these drying racks and they'll put them outdoors. Now they live in
Mm.
Phoenix, they live in Phoenix, Arizona, so it's hot almost all year round. They get great sunlight. So as these mushrooms are drying, they're also producing a lot more vitamin D. And
That's really cool.
yeah, on that subject, I personally think that any mushroom that is grown outdoors in the natural setting is going to have higher percentage of those beneficial compounds that we're
Yeah.
looking for. So nature does it best. Nature understands and knows the chemistry that it has created, whereas we
Mhm.
come in and only dissect the odd bits and ends that we think we understand. And so for those of you that are interested, if you can ID and collect some of these medicinal mushrooms and create your own products, especially if it's wild forage mushrooms from Texas, those potentially are going to be more effective than the products that you're purchasing that are from online, that are grown from in a lab setting. So I'm not trying to knock on anyone's companies or products, but as a consumer of mushroom supplements, I found that there's a difference in mushrooms that are typically cultivated versus the mushrooms that are sourced from a natural setting. And so I really encourage everyone to make that comparison. If you have the opportunity to compare something that's been wild foraged to something that's been lab cultivated and see what
Mhm.
works better for your body.
Yeah, yeah, that secondary metabolite process that you had mentioned is super, super important. Like even the medicinal compounds that are created because the environment isn't sterile, the mushroom
Mhm.
and other kind of fungi within that mushrooms, like endophytic fungi, like it's creating
Mhm.
all of these defense secondary metabolites that then like end up benefiting us in different ways, like both medicinally, those compounds that are created by the endophytes are pretty cool. And this is like
Yes.
you can go down some fun rabbit holes, like reading about secondary metabolites and endophytes. It's a fun place to go. And there's a lot of cool research happening in that space too.
Yes. I definitely recommend checking it out. That was a great question. Thank you for asking that.
Yeah. Irina, so you asked, is it that possible in Texas heat only, or, only in the winter outside? So I can
Um,
speak to this one just being in
okay.
Texas. Yeah, like, you know, we've, with this block program that we have, like there's like probably like 4 or 5 months, and there have been some years where we're still finding that like growing mushrooms, with cultivating mushrooms like outdoors with low tech, no tech methodologies, you can only do it within like a small window. And
Hmm.
we have a really dry, we've had 2 years of this drought now, so it's going to be another dry fall, and you need the real nitrogen charged rainfall to get like a nice fruiting. Uh,
Hmm.
and so uh, it's pretty difficult to simulate. And it's a good thing that we have the ability and the technology to do indoor cultivation. But when you can, expose those mushrooms to UV light, either synthetic or outdoors.
Hmm.
But it's kind of cool to see grow setups like, in Pennsylvania, or even like, would you say, I think you toured the what's the big farm in California where they're right on the coast and they do a lot of outdoor grows?
Mm,
Um,
what was it?
it's like, is it Southwest? South? No, I forget the name. It's
Far
big,
west, far
Far
west.
West, Far West. Yeah. Do they do some things like growing outdoors?
Some of the larger farms will have what they would like consider an experiment plot. Well, they'll do cultivation, but majority of them are typically doing R&D experiments or just kind of for show and display for tours, not so much on the commercial setting. Now, up north, there are a lot of companies that I know that do get into the outdoor cultivation for commercial purposes, but not too many down in Texas because there's just such a short window to grow mushrooms outdoors.
Okay, we'll take a few more questions and then we'll say good night. So Jane asks, what will part of the weekend festival be recorded or live streamed?
So unfortunately, I don't have the live streaming add-on with our venue option was very expensive, so I don't have the capacity to do live streaming. However, I will be recording each one of the talks, and the goal is that they will be uploaded to the Oklahoma Fungi Patreon and eventually the YouTube channel. The only reason that they're not going to the YouTube directly is because of like I mentioned earlier, our YouTube was shut down and I don't want to upload the content to there just for it to get shut down again. I found that Patreon is a great way to keep that information protected, but also accessible for those who are really interested.
Okay. So Greg asks, what the fungus in BC, Canada does growing outside in a hoop house. And
Yes,
he did a mentorship there. Oh yeah,
that is
Greg.
awesome. That's really cool.
Yeah, like there's like the best climate, I'm sure, like a good part of the year. Okay. Yay, everybody. Jacob, I will see you in a few days. I'm so psyched.
Yes. Thank
Yeah,
you
so we
so
got,
much, everyone.
yeah, we'll see you all soon. And Jacob, we'll see you soon in Oklahoma. Hope to see everybody else, a few other folks up there. So we'll be signing off. Good night, y'all.
Much love, y'all. Don't forget okmushfest.com or Oklahoma Fungi on Instagram. See y'all there.